Hello out there. Welcome to Let's Get Oral, where we explore together the landscape of the oral cavity, the spit, the microbes, all of the tissues, and how they're working together to create a whole ecological system that is, bar none, most fascinating. Please join myself, Camille, and my dear friend Jonathan as we jump into the oral cavity together. Today at Let's Get Oral, we have with us Dr. Purnima Kumar, who comes from the University of Michigan, where she is chair of periodontics and oral medicine. When I met her, she was still back at Ohio State University exploring microbial ecology as a periodontist. And what she's uncovered, she's going to share with us today, but it has us looking in the oral cavity with whole new eyes. Instead of looking at single species as pathogens that hurt us, she now has us appreciating the oral microbiome as an ecological niche that we need to look out for and protect. So without further ado, let's get started. Let's get oral. So my first question to you, Dr. Kumar, is can you share with us how you came into the oral microbiome research?
Oh wow, you take me back quite a bit in time,
All the
Camille.
way. All the
all,
way.
all the way where it all began. So this is really funny. I did I ever tell you I'm a thrice born periodontist? No, I got my
No.
first periodontal training in India, and as part of my perio training there, it was a master's degree program. So we did a master's project on Ken Conman's study about the pregnancy microbiome had just come out, oral microbiome in pregnancy. And so we kind of replicated that study. It was great. We took samples from women in 3 trimesters and cultured them. I learned about transport media and culturing and cultivation and all of those things, and did that study. So that was my first exposure to research. Did that. It was a little study. Didn't know anything about publishing, but learned about statistics, all of those things. And then I moved to my second training in England, which I didn't complete, but I was part of it, and that's where the research bug really bit me, that you can actually do stuff like this. And then I came to do my PhD at the Ohio State University, and the time when I started my PhD was when sequencing was really beginning to take off. It was the time of PCR. DNA was king.
I wanted to pop in real briefly to say that for us non-science people, when we talk about sequencing, we talk about this seminal moment in microbiology after we learned about DNA and the genome and how to sequence a genome. This really allowed us to examine samples of bacteria to identify new species, new genomes, to better understand the repertoire of bacterial that were growing in any given space. And so, with that discovery and the tools that came with it and the expansion of the technology, this allowed us to finally see the ecological system that was the microbiome. So back to Purnima.
Right? DNA was king. Everything was DNA. 16S, Phil Hugenholtz had just, and Carl Voss's Tree of Life had come out. So, and David Relman had just published his big paper on the subgingival microbiome in sequencing, and Bruce Pastor had come out with his J. Back paper. So this was really, really exciting times. And I walk into my lab, and my PI takes me by the hand and walks me to a corner in the lab, and he says, "Look at this. This is what your research project is going to be based on." And it was a single capillary sequencer. Literally one little thin coil of copper that was wrapped and wrapped and wrapped around a machine. And yes, you would spend all evening. You would start at 2 in the afternoon, and by 8 in the evening, we would finish isolating DNA. And you'd have this beautiful golden globule of this filmy white material at the end of your test tube, and it was great. You loaded that into the capillary and you left. And the next morning, if you were super, super, super lucky, you got 780 base pairs. 780 base pairs of sequence. Think about it.
So how did you go from this really basic rudimentary science or exploration to discovering TM7, for example, and really creating a foundation of ecology in the oral cavity?
So from there we went to the next level, the explosion, right? This is what we thought we went to 96 capillaries, and it was like the world had opened up. And because you had 96 capillaries, you couldn't literally sequence everything. So we cloned out the 16S gene we cloned it out using the vectors, cloning vectors and plasmids, and then we sequenced those clones. So that gave us the first cloning and sequencing. And so for every sample we did 96 sequences, and oh my god, we called it high throughput sequencing. And just as we were finishing the last, that was my PhD, and I wrapped up. And as the last publications of that started rolling out, we started pyro sequencing, 454 sequencing that came in. And that, right, that completely changed. So now we were getting 1,000 sequences, and 1,000 seemed to be, oh my god, what, tenfold increase, right? So first was a 100-fold jump, and then there was a tenfold jump, but it was truly an exponential jump. And even before, literally before I had even paid for my last 454 sequencing, I hadn't even finished paying the invoice on that. We moved from there. Really, I'm not kidding you. These are all true facts. That I took 5 years to go from, I took 1 year to go from one capillary to 96 capillaries, and 4 years from there to go from 96 to 454, and then 5 months to go from 454 to any other kind of sequencing. So literally the explosion in technology paralleled the explosion in our own research methods. And as these worlds opened up to us, and as we realized, one of the things that I realized very, very, very early was that you can use the sequence information both as a telescope and as a microscope. So it allows you to really, like the Hubble telescope, get the big picture view, right? If you collect enough data points. Remember how this telescope actually let in light for 4 months before it saw those distant galaxies? The new pictures we're getting now is 4 months of light accumulating inside the Hubble telescope for those images to come out, right? Exactly like that. So if you collect enough data points, you can use this as a telescope. You can get enough information about a large community of individuals, about large systems that happen, how different groups of bacteria and genes come together to form a community, a system. Or then you could, use it as a microscope. You could literally go deep down, take one little gene. For me, at the beginning, it was a 16S gene, and now I've realized, I'll talk to you about that in a minute. But then you can take one little gene and say, how is this gene a marker of something across populations, right? So you can look at it at the microscopic level. So that ability to zoom in and zoom out starts giving you the complex three-dimensional picture. You're not looking at it anything more two-dimensionally at this point. It's completely three-dimensional, and you're able to construct these complex landscape type images. And that three-dimensional view is allowing us to study ecology in all its glorious technicolor.
I know that you and I have been circling the oral health research category for some time, but one of the moments where I got to actually watch you in action give a presentation with one of my coworkers, we were really piqued by your research. And I think at the time you had kind of come out with the notion that in the gut, we have seen this ecological aspect of the microbiome, and they push a diverse microbiome as being healthy, but you had the opposite finding. So tell me about that. What was surprising about this for you?
Yes. Okay. So we drew a lot of conversation, I'd say, around that topic when we published that research. But here's what we found, and I'll tell you how it started and how it's developed and how everything we find keeps reinforcing that belief, right? So we found that there's a small group of bacteria that come together um, when you're healthy, and they stay together and they work together and they have these intense collaborations with each other, not just for good neighborliness, but also in terms of nutrition support and structural support and all metabolic support and everything, and just joining forces together to speak to the immune system, because these bacteria live in your body with the consent of your immune system. Your innate immune system says, I allow you to be here. And that is why your microbiome is different from my microbiome, even though we are women of a particular age group and we've lived in the, we've eaten the same kind of food and everything, but still there's these unique signatures that distinguish us because of the differences in our innate immune surveillance system. So these bacteria live here as guests. I don't want to use the word guest. It's not the word guest. It is whatever. As your partner, that is an active partner, but at the mercy, at the invitation of your immune system. So, so when you think about that, why would your immune system let or encourage this community to live in you? The first thing is these bacteria do what is known as smart work, right? There's a small group of bacteria, easy to maintain, easy to communicate with each other. And so we looked at species and we said, oh, there's just a small group of species when you're healthy, and it's repeatable. And the small group of species is present in most individuals, if not all individuals. So then we said, okay, it's species. Then we started looking at genes. We started expanding from the 16S sequencing that I was talking about to whole genome sequencing, looking at all genes that are present in a community of bacteria. And we found that these bacteria are present there for a reason. They do what is smart work, right? They literally use Krebs cycle, oxidative phosphorylation. They use oxygen in your body to break down small molecules of carbohydrates and proteins, very simple molecules. So they really generate a ton of energy that they use for their bodily function, so to speak, to regenerate their cellular organs, to transport nutrients, to replicate, to divide. They use that, and they're quiet. They're like great Airbnb guests. They come in, they pay their rent, they don't throw rager parties, they're quiet, they don't stay up after hours, they don't clog up your sewer systems. They do their thing. They're smart. And so your immune system loves them. And apart from that, humans are very selfish. Humans are very selfish species. So we're very self absorbed. And so we want things that benefit us. And when you look at the oral microbiome, there's some beautiful research about how this microbiome actually benefits your vascular supply, how your epithelial cells, which is your epithelium, your lining mucosa, that is your most powerful immune organ in the body. And these bacteria are constantly talking to these cells, these lining cells, and telling them how to maintain the junctions between them. If you don't have these bacteria talking to you, you get what are known as leaky junctions, and that causes its own host of problems. So these bacteria are helping you, and they are being nice to you. So that's all it needs, a little tiny community of bacteria that's self-sufficient, self-feeding, self-taking, and by the way, they're taking care of you as well. So that's a group of bacteria that comes with very specific functions. And that's why we don't find that common core, right? Because the same group of bacteria, what you need is a common group of functions. There's a specific group of bacteria that can provide those functions, and they don't have to be the exact same between you and me. So you can have a different suite of bacteria, and I can have a different suite of bacteria. But when you look at the genes that these bacteria bring to the party, it's the exact same. So functionally, these communities are very similar, right?
I really appreciate you, yeah, bringing to the table all of those functional components. Do you think that early on, as we acquire this oral microbiome, we set the stage um, with our immune system in coordinating that kind of fixed relationship? Do you think that there is room for uh, change over the course of our lifetime? Or do you think it's fairly static?
Okay, that's a great question. So I'll tell you what we've found, and this is based on several different types of research we've done. We started by asking a very very simple question. We said, does the microbiome change over decades of life, right? When you're first born, before, and we looked at teeth. I mean, I'm a dentist, so for me, teeth, as you start with you start with no teeth and you end life with no teeth, right? So then you get
Yes.
in between, right? So life, the full cycle of life is no teeth, 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 no teeth, right? So you go through the cycle of life. So when you think about it that way, we said, okay, let's use the dentition state as a marker of does this bacteria continue. So what we found is this: there is a very core group of bacteria that you acquire even before your teeth come in as a baby. So, and these are acquired within 1 day to 1 month of life. Within the first month of existing on Earth, you have acquired a core group of species. 95% of these bacteria are carried through into the adult dentition. I'm talking about 18 to 20-year-olds. So it carries into the adult dentition. Then you get a second core group of bacteria that only can be, need teeth to be present. So they only can colonize tooth-like surfaces like Streptococcus mutans, Porphyromonas gingivalis. All of these organisms really need an abiotic tooth surface to colonize. And you get that second suite of organisms when your first baby teeth come out. And those children in our study were 6 to 9 months of age. So before you blow out your first birthday candle, 90%, 80 to 90% of your adult microbiome is established, is done. But there's room for change. That 10%, and it's not 10%, oh, these are the rare species and the rare taxa. There are actually some very robust dominant bacteria that can, numerically dominant bacteria that can come into your microbiome later on. So there is a 10% leeway where lifestyle can change these bacteria and your practices and things like that. But essentially, the foundations of your adult microbiome are set before the first year of life. So familiarity. And then we asked, where are these bacteria coming from? That's an important question, right? So
Yes, yes.
the ones that the baby acquires before their teeth come out are an entire subset of their mother, of the mother's oral microbiome. So the mom is the one who gives it to her babies. So we're seeing that, and we have reason to believe it's through vertical transmission that the mom is, just like she gives her genes, her DNA to the baby, she also gives her microbiome to the baby through vertical transmission, like inheritance. But there's some beautiful work that has been done by Anne Griffin and others that show that adoptive mothers are able to, transmit their microbiome to their children as well. So there seems to be two modes of transmission. Um, and either could be possible, or both could be possible and could be working in conjunction. So yes, there is an early acquisition and it stays on. So yes, it can change over time, but I don't think it's more fluctuations rather than change, I would say.
So, with that said, there is a growing awareness in the dental community around the oral microbiome, and this kind of push to get a chairside evaluation of the microbiome by PCR techniques that you described earlier. How how do you think we can navigate this new tool in practice? Because as you say, there's not a lot of fluctuation. So it seems like we're just kind of doing a bit of a 23andMe just to see who's there and what's going on. Do you think that there's a meaningful application here?
So let me ask you this question. If I said to you, I want you to tell me the health of the Great Barrier Reef, and oh, here is a mill of seawater from the Great Barrier Reef, what would you be able to tell me with that?
Well, it doesn't really contain any of the reef, so I'm not sure what I could say about it.
Thank you. Thank you. So here's I don't want to say this is doesn't contain any of the reef, but here is a medium that is free flowing. It's like, it's saliva. It's secreted as a sterile liquid. It flows through every surface on inside your mouth, on your teeth, on your cheek, on your lip, on your tongue, under your tonsil. It goes everywhere. And we know that the mouth is a set of habitats. It's a set of habitats. It's like, you have the tongue, which has its own microbiota. You have the cheek, which have their own microbiota. You have teeth. I just told you teeth. Some bacteria can only live on teeth. So now this is a thing that flows through all of these surfaces, and then you take it all out and you spit it into a cup, and you're saying, tell me if this person can have gum disease on that one lower molar tooth. That poor little spit how can it tell you all of that, and how would you be able to find out that? The trouble with using those kinds of global diagnostic tools, the challenge, I don't want to say trouble, the challenge is that unlike IBD or pick your C. diff, pick your poison, right? One of those gut infections, diseases in the mouth. Not every tooth in the mouth is affected by disease.
Of
There are,
course.
even in a person with disease, they have healthy teeth. So it's very site specific, and it's very random. You cannot say always, oh, it's the molars that are going to be affected, or it's the front teeth that's going to be affected. There's no way of making those. Yes, there are probably a higher probability that a molar tooth would be affected by gum disease, periodontitis, and you know, the chewing surface of, or the surface, the between two teeth on the molar might get the first cavity, things like that, the interproximal space as it's called. But there are no guarantees. The probability is high, but there are no guarantees. So, and you don't even know that all interproximal spaces or all molars will get disease even in a person with disease. So you're talking about a highly site specific, extremely random event, and you're taking a global fluid that would that you're asking it to be diagnostic or prognostic of a very site specific
Right. I
disease.
think we can all agree that the diagnostic capacity for something like COVID is more realistic, but maybe not periodontal disease or active infection.
I'm sorry, I lost you there for a minute.
I saw that. It's okay. Um, well, this whole talk about the more fixed community of bacteria all doing very similar things ecologically in your mouth and in mine, despite the different or variance in species level community. But there is this functional similarity. So could we kind of move into the idea of metabolomics and kind of ask metabolomics to give us that chair side benefit, or what are your thoughts there?
No, it's entirely possible. And we actually just completed a study where, we've always puzzled over this question of why does someone with a lot of plaque have little inflammation, and why does someone with really an absolute clean mouth have a lot of inflammation, right? So the high responders versus low responders to plaque. And this time, exactly like you said, Camille, we said, we're not getting answers from species, we're not getting answers from cytokines. Let's go look at metabolomics. And we do find that there are very distinct metabolic profiles that are associated with people who are high responders versus low responders. Again, to me, yes, there may be a global response, and metabolomics is at a stage where you need higher sample biomass to be able to get those kinds of answers. But the question is, for a site specific disease, if you want to ask, does someone um, have disease in that place, then you might want to use samples from very specific sites that you think might have disease, right? And there's always the question of how we currently diagnose disease clinically. You know, as a
Yes.
diagnostic marker, you cannot beat the periodontal probe. It is a
Yes.
simple metal instrument that costs $10. It is sterilizable, and it gives you an answer within 20 minutes, which is what it takes to probe the full mouth. And it tells you with great accuracy not only where there is disease, but how much of disease there is, and what is the nature, the type of the disease as well. So you get a lot of information from that clinical diagnostic tool. So I would look for prognostic markers: who is more susceptible to disease, who has
Yes.
a higher risk for getting disease. The 23andMe, if you could use it not to determine um, what is in the past, which is a historical, thing of disease, a historical record of disease, which is what the periodontal probe is. When you measure disease with a periodontal probe, you're measuring an incident that happened several months ago or several years ago. But to say what futuristically who can have disease, that's
Absolutely. I think Ken Corman would be right there with you on this exact issue. And I think that there are a lot of pieces we can put together now to give us some of that insight. how we deploy it, I think, is a little bit more complex. And I don't want to get too political, but I do know that there are a multitude of factors going on at play there. Um, but so when we think of these kind of microbiome diagnostics, we're also looking at the onslaught of probiotics being deployed to help the ecological health of the oral bacterial community. And I myself conflicted because there's some really compelling articles, research articles that have shown a benefit. And so I'd love to get your thoughts on what you think about the probiotic um, as a cure-all, or even as a
Camille, Camille, what
yes,
did I just tell
yes.
you? What did I just tell you about why? Why do you
Well,
even
I
think
love
that?
I love it when you really spell it out, because then that gives us a really solid picture.
So think about this, right? So think about this. Let's say I drink a glass of water now, right? How many of those bacteria do you think will stay on in my mouth and colonize?
None. I don't think anything will.
Exactly, right? Think about this. This is the mouth. It is the gateway to the body. It is not the gut. It's not a protected system. It's not, protected at both ends. It's not the middle ear. It's not the nose. It's that in your face community. Everything and his brother goes through the mouth, right? So the oral microbiome truly are the gatekeepers of your health, if you think about it that way. This is a juggernaut community. And why was this community formed in the first place? What allowed it to be formed? This was formed before your first birthday. And remember how long it takes for your immune system to mature, at least 2 years. So, this is a community
Yes.
that was established early on in age in your life, and it's telling your immune system, listen, brothers, we're your friends here. We're all going to be living in harmony. We're going to be friends together. We're here to protect you. We're playing a role in your life. And by the way, you know us as friends because you saw us when on the other side of the maternal placenta. So there's this lovely research that we're getting ready to publish. It's coming out right now, where we found that the oral microbiome of the mother always translocates to the placenta. I don't want to say oral microbiome; it's the wrong word. Bacteria from the mouth and bacterial products and bacterial genes, broken down bacterial stuff, anything whole cells as well as broken down material, move from the mouth through the serum into the placenta in all cases, whether you're having a healthy child, whether you have delivering preterm, it goes there. Now,
Well,
what
that
what
wouldn't be surprising because of what we've been finding about the blood microbiome, right?
right? Right? So,
Yes.
and we're tracking it through the serum. So we went from the mouth through the serum to the placenta, and yes, and yes, right? So that the track record is being established. And so, what we believe is that the fetal immune system on the other side of the placenta is seeing the Treg cells. They're seeing these bacteria and bacterial genes, and recognizing them as friends. So when the baby is born and comes out, and the mother picks the binky out of her mouth and puts it in the baby's mouth, these bacteria are accepted by the baby's immune system as friends. So that is how these bacteria are allowed to colonize, and that is why the mother's oral microbiome now becomes, the source of the baby's oral microbiome, right? And then these bacteria stick on there and say, hey, we've known each other even before we were born. We're friends in neutral. Let's go through life together, right? So when you think of a
I love
system
it.
that's set up like that, it's evolutionarily set up because then we know as humans the baby is going to pick up everything from dirt to everything and put it in the mouth. The human is going to do that. Other influences are going to come in, and this is a juggernaut that cannot be moved, should not be disturbed or disrupted in any life. Then what is the point in bringing a foreign species and saying, hey, you know what, if I drink a glass of water, don't let the bacteria from the glass of water colonize you. But if I intentionally take a little pill filled with blue bugs, let them in and be part of your community and live in harmony with them, right? So to me, that it might work in a system that has no microbiome and you're resetting it. So It's great for diseases like IBD and C. diff and all of those where there's nothing, there's ground zero. You're literally rebuilding everything up from ground zero. But in a system
Right.
that's already has a lot of, I mean, think about the mouse studies that you did, Camille, back in the day. If you wanted to
Absolutely.
colonize a mouse with P. gingivalis, what did you do first? You brought ground zero by feeding antibiotics in the
Yes.
water, right? So you killed everything off before you put P. gingivalis into that mouth and asked it to grow back. So yes,
Or the
these things,
ligature model. Yes, you have to really, really put it in there and make it
right,
last
last, and you had to
with a
keep on
lot of
refeeding.
inflammation.
You're right, right.
Right.
So, and you said the right word, with a lot of inflammation. So you have to set the stage for that bacteria to take root and grow, right? So
Right.
I would argue that to maintain health, you want things that will set the stage for your healthy microbiome to come back, which to me would mean prebiotic and not probiotic, right?
Indeed.
It's an ecosystem.
Yes,
How do you change an ecosystem? By changing the ecosystem, not by changing the people who live in the ecosystem.
absolutely. So prebiotic or just even maybe healthier foods potentially.
Functional foods. Hey, food is food
Functional
as
foods.
functional foods. Totally all over it. I love it. I've done some work on it, and we're getting ready to publish that. Black raspberries in
Oh,
smokers.
excellent.
Oh, it's
Oh,
beautiful work. I'm so in love with that. So Ohio State,
interesting.
yes, Ohio State
Okay, I look
University.
forward to seeing that.
Yes, yes, yes.
I do have to say, so we do know that there are functional components that the microbiome is giving to us so that our bodies allow them to sustain their community. And so we know that they're part of our metabolic health, for example.
Right.
And some of these mouthwash studies are really jaw dropping to show that the nitric oxide production is really disrupted by just the application of an antibacterial mouthwash. Um, and yet, at the same time, I felt compelled to kind of love the COVID studies that used mouthwash to like mitigate some of that viral load in the mouth. So I could see how um, chairside practitioners may be a little confused about how to deploy these mouthwashes, given that now our much better understanding of the oral microbiome.
Great. I'm going to again give you a little bit of story background on the nitric oxide studies, right? So when you think
Excellent.
about it, we've always the dogma that eating green leafy vegetables is good for your heart, right? That is one side of the
Yes.
picture, and the other side of the picture is that people with more severe and more extensive periodontitis, gum disease, have poor heart health, or have oral bacteria can be found in their atherosclerosis. So there is one side of the story where good bacteria can keep your heart healthy, and the other side of the story where bad oral disease can contribute to heart disease. So we'll focus on the health side, right? So when you eat green leafy vegetables, it's supposed to improve your heart health. Well, eating green leafy vegetables is important because green leafy vegetables contain something important called nitrites. Well, our body doesn't have the enzyme that is needed to break down these nitrates. You need nitrate reductase to break down nitrates into nitrite and nitric oxide, which our body doesn't have. The humans with the best will in the world don't have the gene that codes for it. We rely on bacteria. So the oral bacteria, and again, I'll talk to that in a second, but oral bacteria have this um, enzyme that can break down green leafy vegetables and release nitrates from that. So once you have nitrates released from that, they enter your saliva, your salivary glands, and they enter your bloodstream, and where they're broken down further into nitric oxide. And we all know, what does nitric oxide do? It dilates your blood vessel, and by dilating your blood vessel, it lowers your blood pressure, which is how eating green leafy vegetables is important for heart health. But that is just one part of the story. Eating green leafy vegetables when you have gum disease and bacteria that cannot break down your nitrates is nothing. It just sends them down the tube. That's all it does. So it doesn't do anything for you. So it's a combination of being having healthy gums, having healthy bacteria that are capable of breaking down, and having a healthy diet that's full of green leafy vegetables. So it's a combination of these two things, right? So when you're absolutely healthy, when you have oral health, do you want to destroy the bacteria that live in your mouth by using a mouthwash that will indiscriminately kill or mechanically remove these bacteria?
I, well, I like the idea of using something like chlorhexidine around, therapeutic moments of trying to mitigate periodontal disease, but perhaps maybe not for months on end.
Or even when you're healthy, why at all a mouthwash? What
Indeed.
benefit would a mouthwash offer someone who has no gum disease, visits their dentist every 6 months, gets their annual prophylaxis, is in the top shape, right? Why? That is one question. So that is the fundamental question that I would ask. It is not.
But wait, what if we have our diagnostic? We know who's susceptible and they're prone to perhaps developing periodontal disease. Then what?
Then why would you indiscriminately kill every bacteria in their mouth? Why are you
Okay,
sending them further
just
down the,
checking.
right? So that is a
Yes,
rabbit hole that you don't want to enter, right? Because you don't know what's
yes,
coming up, right? You can take
yes,
down a community, but what's going to be replaced with under those kinds of constraints, right? So
yes,
why would you? That is a direction you don't want to take. So don't fix it if it's not broken.
yes.
Because susceptibility to disease is, again, you know as well as I do, if not better, that it's a whole ball of wax. It's like a hairnet. There's so many threads there that no single factor can drive susceptibility to disease. You can carry the genes that make you susceptible to disease, but go through an entire lifetime span never getting the disease. So again, it is those factors that trigger onset. So controlling the environment is the best way. And by environment, I don't mean using mouthwash in the mouth, but the environment around the body, the food, the lifestyle, the habits, the obesity, the stress, the cortisol levels, all of those things around that all the factors that drive a healthy microbiome. So that, I think, is the most important thing. Now, when again, this is like COVID is like the IBD and the probiotic story, right? In an extreme case where you're looking at a respiratory pathogen, for example, and we did the COVID study. So the reason why someone who is so anti-mouthwash as me would even do a study like that is, think about this: you have a respiratory pathogen that at that point in time was literally, to get it was a death sentence, close to a death sentence, at least a hospital stay, right? So the question was, how do we decrease transmissibility of this, and how do we reduce the oral levels of this? Again, I want to just say this: oral carriage rates. I'm not using colonization, I'm not using the word invasion. Carriage, how much is present in your mouth and in your nasal, in your oral pharynx, this part of it, your throat area? How do we reduce that? And we
Right.
found that mechanical rinsing, it doesn't have to be. We used everything from saline all the way to iodine to chlorhexidine and hydrogen peroxide. We found that all of these will comparably reduce your COVID viral levels in the mouth, the carriage rate, for 15 to 45 minutes. So it does do that. So in extreme situations like that, where you want to abruptly reduce the level of something, it doesn't keep it away. It's not like, oh, you can use this and you're safe. It's on the safety shield for the next 6 to 12 hours, right? It
Right.
doesn't do any of
Right.
those things. It is you.
I, w-
you
I was thinking of a good application for something like that is, when you are living with your family members who might be more susceptible and you have it and they don't, and little things that you could do, like if you know you're going to be interacting with them, maybe just do that first,
Absolutely,
so that
absolutely,
you minimize the risk, right? But
absolutely.
again,
Rinse your
not
mouth. Exactly. Literally, just rinse your mouth out, wash your hands, put on your mask, and go talk to them, and have them mask, because two-way masking is better than one-way masking, right? So
absolutely.
exactly, as part of a multiple step approach to disease and infection transmission control, it's great.
Yes,
But when
agreed.
you're
I
living
just
your
I
best
I was
life,
afraid. when yes, when right, exactly, when you're living your best life, you don't necessarily need to go through all that. I was just afraid when the studies came out. I was excited, but at the same time I also didn't necessarily want people to pick it up as though it was hand sanitizer, and really taking it a little too far, because I felt like
I
that there
know.
was a that we could do a lot better of communicating these nuances, at least with the dental profession, so that there's a large awareness to kind of move forward cautiously and strategically. But since we're on the subject of COVID, I have found it to be really enticing, to see some of the periodontal research coming out with COVID that is showing, increased infection, salivary viral load, and do you think it is because of that oral bacterial community that is involved with periodontal disease that's allowing that transmission, or just um, the fact that these people are traditionally or commonly have multiple systemic inflammatory factors? I love this kind of idea of the immune system and our mouths as a gateway, and that this COVID periodontal disease interaction is somehow meaningful. What could you say about that?
I'm going to say we don't know much, and it's easy to take a button and sew a vest on it, and make, the circumstantial evidence is so high right now, and we can easily be led down different paths. So with a very, very healthy dose of caution, I would say multiple mechanisms are possible. The first one is the comorbidities, right? What causes
Yes,
periodontal disease, what contributes to severity of periodontal disease can also contribute to getting COVID, transmitting the virus, all of those things. The second thing is that we know viruses cohabit with bacteria, and there's a very strong interkingdom collaboration between them. And I'm not talking just about the viruses that live inside bacteria, like the bacteriophages, but truly
sure.
just human viruses, like the herpes virus and the cytomegalovirus, the one, the common cold virus. We've known for years, for decades, that these things hang out under your gums, that they live there, that they actually have a very cooperative relationship with the bacteria. Exactly what those relationships are have not been completely mapped out, which means we cannot say how having certain groups of oral bacteria can enhance your risk for also carrying the SARS-CoV-2 virus. But there is evidence. There is very circumstantial evidence pointing to, yes, there can be. And of course, the periodontal disease is a chronic inflammatory condition, and who is at high risk for COVID, SARS-CoV-2? People with chronic inflammatory conditions. And hey, here's one more chronic inflammatory disease. So, that, and about two-thirds of the population has it. So just by odds of statistical analysis, you're going to always include a few people who have both SARS-CoV-2 and periodontitis, and suddenly it looks like they correlate with each other when it could just be
It's
a
true.
random correlation as well, right? So we don't know enough. We know so little, and we should not rush to any sort of conclusion with that. But
I do love that you brought up the virus commingling with our oral microbiome, because I think that that is an area that I would love to see a lot more development as far as research. And I do see that there are some researchers taking on the virome, the oral virome, but somehow not enough. Are you
no,
taking that on?
yes. So not the virome as such, but just, we've expanded our search of the microbiome to include all members, fungus, viruses, and we started this with kind of a clean playing field. We started with placing an implant, and then when you get an implant, which is literally just a screw that goes into your jawbone and becomes the root of your tooth, the fake root of your tooth, you close the gum over it and let it heal for 3 months, and after 3 months you expose the implant to the mouth, to the oral cavity, right? It sticks out, and there's a little trainer crown on top of it. So that trainer crown starts accumulating bacteria, so you can literally study bacteria from day zero all the way up until whenever you want. And so we've been doing the study, we've been following the development of this community of bacteria that live under the gums right next to the implant. And we're finding there's a very robust colony of viruses that are living there. They start from day two and they build up and they have these beautiful collaborations between bacteria. And this week only knew because we did the whole genome sequencing as you know, and we look at these communities and we're like, hey, they're there. So they're not a marker of health. They're not predictive of disease. They're just there. Yeah.
I have so many questions about that. That is fascinating. Does the difference in materials affect the study? Were there different posts?
So this study, because as we did the study, there was all this chatter in the background about, oh, it's material, it's this. We cleaned it up for one type of implant, one brand of implant, one material. Even within the same brand, there can be multiple configurations of implant. We
Yes.
can implant configuration to be the same, almost the same tooth in every person. Very very tightly controlled, totally non-generalizable study, but really we needed that.
The pilot, yes.
How every paper you always has say our findings are generalizable to the larger population. This no 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 no. This is a very tight group, but we needed that kind of homogeneity to understand what is going on, because with human studies, that's a big thing, right? Humans are so
Yes.
variable and human behavior is so variable that the noise drowns out the signal, and we didn't want that. And that is how I think that was a big strength of the study, that we're actually able to detect this in multiple different humans from different walks of life with different practices, with different everything. But there's a common thread that's running through all of them as their implant microbial communities grow and mature and develop and stabilize.
Very nice. I love it. That's fantastic. I do want to see more materials, but I think they'll probably have a similarity that
No, no, I'm
is
sure it will be
generalizable in
covered.
the future.
Hopefully,
Yeah,
if
yeah,
not, then we'll just that just opens up avenues for more research on each material, right? But that's also
absolutely.
an important finding, and we need that will be the next step, is to say, hey, what happens with different materials? So those studies will come out. But this was something we had to do because nobody had done that. And again, it was just living on faith and sunshine and saying, oh, maybe this is how it happens, and therefore we will treat an implant like this. And I'm guilty of that. When I do implant surgery, I'm like, I think I know what I'm doing, but I'm going to tell you I know exactly what I'm doing, and I know it's going to work for you. But that's my surgical side, and then my scientist is sitting like that little thing on my shoulder and saying, "You don't know, you don't know, you're lying to the patient, you don't know, you don't know."
Yeah,
So
yeah, that's true. I'm glad that you have the passion to move that research forward. Last question on the implant. So we all know that there are uh, slow, low responders and high responders with regard to perio and gingivitis. Did you see that kind of come through in this study?
not in this study, but we did another
Okay.
study, and I'll tell you about it. So, with gingivitis, you know how we always say gingivitis is reversible. Brush and floss your teeth and use whatever you want, and
Yeah.
and gingivitis will reverse back to normal. With mucositis, peri-implant mucositis, which is the analog of inflammation around the implant, you get two shots. You get two shots at getting peri-implant
Mm.
mucositis, and then the whole host bacterial interaction, that immune bacteria interaction that we were talking about, doesn't reset
Yeah.
itself. It doesn't reset to
Ah,
normal. It's right. So the train wreck is on the tracks after two. You get two shots. That's it.
wow. Do you think
That's
it's
it.
because of the missing periodontal ligament?
Could be multiple reasons, right? The the,
Yeah,
the
okay.
way the, right we again, I don't want to even say it's multiple reasons. I'm saying it could be because who knows? Who
Yeah,
did the study? No one. We're just
true.
starting to tease this out, and it's painstaking, it's slow, it's question after single question, because you can't ask multiple questions in the same study, because then that'll, you'll always get an answer based on false positive rate. So you have to test each one, and then each study opens up more questions than it answers, and that's what they say,
Yes.
right? The hallmark of a study. But
I do have this dream of getting practitioners updated on the current kind of frontier science, but how to make it applicable, or is it just for their awareness? I think that there could be a marriage there, but I'm not exactly sure what that looks like. Do you have a vision of that?
Um, Practitioners, yes, and the problem with human studies is that it all relies on how granular and how accurate your metadata is. Anyone can collect a sample. I can have every patient spit into a tube and bring it to you. But did I take accurate data on the patient's diabetes status, smoking status, vaping status? Did I ask questions
Yeah.
about their BMI, their diet, their food, their age, their gender? Oof, how
Toothpaste.
many times they've been pregnant? What toothpaste they use? How many times they brush? How many times they floss? Right? What type of brush they
Yes.
use? Right? So
Yes.
those kinds of, and then the question is, what other data did I not ask you? What should you be telling me that I
Yes.
didn't ask you?
Yeah.
That as well, right? Alcohol use, marijuana use, recreational, medicinal. Um, we, we need, and that kind of granular data can only be done under the circumstances of an intentional study, right? If I went to a
Right.
private practice and said, give me all this data, they can give you what is in their medical history, which is a self-reported medical history, and their
Right.
demographic, which is again which can be a little more accurate, but how many times has your dentist asked you what your ethnicity is? Was that ever recorded?
That's a great question. I don't think so.
I would say no, because I've not asked any of my patients, and I've not seen that on any of their. Maybe I have for some, but gosh, you got me thinking. I was, as I said this, I'm thinking to myself, God, do I record that data? Because I certainly don't use it in any way. The
Right,
medical history,
right.
the demographic information gets preloaded into your EMR, and I'm thinking to myself, did I ever bother to look at that and make a meaningful, use that as a meaningful data point? I'm going to say no.
Right.
So,
Yeah. Yeah,
so,
interesting. I mean, you do for trials only because you want to fulfill kind of the population bias or
or
what
you
have
want to
you.
actually understand if there are differences between, right? So it's intentional. But if you're looking at a practitioner and collecting data from a private practice setting, it is how much granularity of metadata that you can get. The clinical data will be beautifully available to you. I have no questions about that. But what
Right.
factors are associated with that clinical finding is, I think, our biggest roadblock.
It- yes, that's really interesting. Well, I see that we're coming up on the hour, so I'm going to ask you one last question, and that is, you throughout all of your articles and all of your writings, you use poetry to accentuate a particular learning. And so I thought, could you share with our listeners maybe a book of poetry, some essential book for their collection that you wouldn't go without, you wouldn't couldn't go without?
That's so funny. If I hadn't been a periodontist, I'd have been a historian. I love history. I love literature. I grew up. My early education was in India. I told you that. My education was in India, and in India, part of our curriculum was English literature, and we grew up with the glory poets. Keats, Browning, Tennyson, Shakespeare. So all of these people were part of my learning. Robert Frost. So we even knew where they came from. So I learned more about the transcendental poets and transcendental poetry sitting in India, way back when. So that was part of my education, and poetry was the just the meter, the ambience, the pentameters, the iambics, all of those beautiful things has always been part of my life, and I love it. I just love how you can express a thought or an emotion, or just the conciseness, right? You can take a whole world of ideology and thought and put it in a sentence, and people read that one line and they're like, yeah, makes total sense, absolutely got it, I get the message, right? I don't need to go any further. That line speaks to me like nothing ever has. So the ability to synthesize incredible amounts of knowledge or understanding of philosophy into a few words, few telling words, And then we write these verbose 15-page, 20-page manuscripts. And I'm like, there has to be that one thing that crystallizes the entire thing I'm trying to say in this manuscript. And I turn to classic literature for that, which is why you have A Tale of Two Risks, or you have Anna Karenina, or now
Yes.
This Man is coming out. So those, because I want the central. And to me, as you will probably notice in this conversation, I can go down rabbit holes and never find my way back out.
I know, I truly appreciate those little golden tidbits because it does help kind of give that broad but tight picture. And it adds a level of philosophy to it that I really appreciate.
And so when we think about it, right? So then I come to the United States, and our daughters go to school here, and I learn Shel Silverstein. Oh my God, what a cutie, right? What amazing, hilariously, like, strikingly funny. Again, he can take a whole joke that you can tell over 5 hours and crystallize it into 2 lines, and you're like, done, done. That's it, right? Or two words, and that's what a kid would read. And then those
Yes.
kinds of poetry that can wrench and actually go into your heart and sit there and never leave it, and make you think and think and think and think and say, I want to learn more, I want to know more. So yeah, all those. So I've started reading Robert Hayden. He is my
Robert Hayden.
Robert Hayden. He writes very disturbing poetry. That's the word I would use for it. And if you haven't read the poetry called Middle Passage, it's about the journey of the slave from the slave boat from Africa to and I've read many history books on slave trade and slave labor, but that kind of non-graphic description is gut wrenching. It leaves it sears your soul. It doesn't stop with just, oh, I have an emotional connection to this. It literally, I think, it just gets into your soul and it stays there. And those kinds of poetry as well. And that the inauguration, Amanda Gorman, and her poetry and the power
Yes.
of
Oh,
hope,
yes.
right?
That was incredible. Yes.
Right. She distilled everything, that every hope that every different person in every different walk of life had, into those 5 minutes. And I was sitting there with my jaw dropping the phone and saying, you, how old are you
Same.
again? I'm like 200, 2,000 years old.
Yes.
Just the age, right? It's so ageless. So yeah, that's why I love classic literature and classic poetry so much. And I keep going back to that well to drink from it all the time.
That's fantastic. Thank you so much for your time today. I really appreciate.
Camille, it's always such a great
Mhm.
pleasure. We've worked together for so many years in so many different formats, so many different circumstances, so many different environments, and it's never stopped being a joy. So thank you for this opportunity. I'm so glad we just got to sit and chat about the two things that interest us most: the human immune system and the
Thank
microbiome.
yes. Yes, it is all I care about, really. And I appreciate that you are still hauling the frontier of this business because you've done an incredible job. So thank you. I'm
Thank
gonna
you.
stop. I'm gonna stop this bus right now, but here. In closing this episode, I just wanted to run through what we've just heard, and that is the kind of evolution of how we've come to research the oral microbiome. Our initial findings were, to look at these pathogens, and then we realized that no, they're part of a much bigger story, and we're still learning at blistering pace what is happening in your mouth and how it relates to our oral health, but also our systemic health, and how the two impact each other. I love that Purnima is so passionate and really heading this whole ecological understanding of the oral microbiome, and I cannot wait to see where her research takes us. But I wanted to reiterate her favorite literature pieces, and that is the classic literature, Shel Silverstein, Robert Hayden, Amanda Gorman. Thank you for listening.